For 40 years, nutritional guidelines in the United States and most of the Western world told people to limit egg consumption. The American Heart Association recommended no more than 3 eggs per week. Dietary guidelines warned that the cholesterol in eggs raised blood cholesterol and increased the risk of heart disease. Doctors advised patients with cardiovascular concerns to avoid eggs entirely. A generation of people switched to egg white omelets and felt virtuous about it. The science underlying these recommendations was wrong. Not slightly wrong, not wrong in a nuanced way that required careful reinterpretation, wrong in the fundamental causal claim that dietary cholesterol raises serum cholesterol in most people, which then causes heart disease. The relationship between dietary cholesterol, serum cholesterol, and cardiovascular disease is significantly more complex than the simple linear model that drove 4 decades of public health guidance. And the research that has accumulated since the original recommendations reveals a picture that is almost the inverse of what most people were told. Eggs are not a significant driver of cardiovascular disease in the majority of people. They contain compounds that are critical for brain function and that most people in the modern diet are chronically deficient in. And the specific form of fat they contain has a relationship with cardiovascular health that is categorically different from what the original dietary fat hypothesis predicted. This is the story of what eggs actually do in the body when you eat them daily, what the cholesterol does, what the choline does, and why 4 decades of dietary guidance missed the most important parts of the picture. The dietary cholesterol hypothesis, the idea that eating cholesterol raises blood cholesterol, which causes heart disease, originated primarily from the work of Ansel Keys in the 1950s and 1960s. Keys conducted the 7 countries study, which found correlations between dietary fat intake and cardiovascular disease mortality across different national populations. The problem with the 7 countries study, a problem that was identified and published by critics at the time but did not reach the mainstream for decades, was selection bias in the countries included. Keys collected data from 22 countries but selected 7 for his published analysis in a way that maximized the correlation he was looking for. When all 22 countries' data are included, the correlation between dietary fat and cardiovascular mortality is substantially weaker. But the hypothesis gained momentum, and in 1968, the American Heart Association recommended reducing dietary cholesterol and saturated fat. Eggs, containing approximately 186 milligrams of cholesterol each, became a dietary villain for the next 4 decades. The metabolic reality is considerably more complex. Dietary cholesterol and serum cholesterol are regulated by different mechanisms. The liver is the primary producer of cholesterol in the human body, producing approximately 1,000 milligrams per day regardless of dietary intake. When dietary cholesterol increases, the liver compensates by reducing its own cholesterol synthesis. This feedback mechanism, called cholesterol homeostasis, means that most people's total serum cholesterol is relatively stable across a wide range of dietary cholesterol intakes. Approximately 70 to 75% of people are what researchers call hyporesponders. Their serum cholesterol shows minimal change in response to dietary cholesterol intake. For these people, eating 1, 2, or 3 eggs per day produces no meaningful change in total serum cholesterol. The remaining 25 to 30% are hyporesponders. Their serum cholesterol does increase in response to dietary cholesterol. But even in hyperresponders, the response is not uniformly problematic. Research has shown that dietary cholesterol in hyperresponders tends to increase both LDL and HDL cholesterol simultaneously, raising both fractions proportionally rather than shifting the ratio unfavorably. The LDL particles that increase in response to dietary cholesterol also tend to be large, buoyant LDL particles rather than the small, dense LDL particles that are more strongly associated with cardiovascular risk. This distinction between different subtypes of LDL particles was not understood when the original dietary cholesterol guidelines were established. The assumption that all LDL elevation was equivalent was wrong. The type of LDL matters as much as the level. The relationship between eggs and cardiovascular outcomes in actual human populations, rather than in the theoretical framework of the dietary fat hypothesis, has been examined in multiple large prospective studies. A meta-analysis published in the British Medical Journal in 2013, analysing data from over 300,000 participants across multiple countries, found no significant association between egg consumption and risk of coronary heart disease or stroke in the general population. People who ate 1 egg per day did not have higher rates of cardiovascular events than people who ate less than 1 egg per week. A subsequent large prospective study published in the American Journal of Clinical Nutrition, following over 100,000 adults for up to 32 years, found that moderate egg consumption, up to 1 egg per day, was not associated with increased cardiovascular disease risk in healthy adults. The exception, and it is a genuine exception that deserves acknowledgement, is people with type 2 diabetes, in whom some but not all studies have found associations between high egg consumption and increased cardiovascular risk. The mechanism is not fully understood, and the evidence is not consistent, but people with type 2 diabetes may have impaired responses to dietary cholesterol that differ from the general population. For the majority of healthy adults without diabetes, the evidence from large prospective studies does not support the claim that daily egg consumption increases cardiovascular risk. The 4-decade-old warning against eggs was based on a hypothesis about dietary cholesterol that has not been supported by the prospective evidence that accumulated after the guidelines were established. Choline is the nutrient in eggs that receives the least attention and deserves the most. Choline is an essential nutrient. The body requires it for a range of critical functions and cannot synthesize adequate amounts endogenously. It must be obtained through diet. It is required for cell membrane synthesis, for the transport of fats from the liver, for methylation reactions critical to gene expression, and most relevantly, for the synthesis of acetylcholine, the primary neurotransmitter involved in memory formation, attention, and muscle control. Choline deficiency is one of the most common nutritional deficiencies in the modern diet, and one of the least discussed. The adequate intake recommendation for choline is 425 mg per day for women and 550 mg per day for men. The average American consumes approximately 260 mg per day, less than 60% of the recommended intake for women and less than 50% for men. This deficiency matters because of what choline does in the brain. Acetylcholine, synthesized from choline, is the neurotransmitter most associated with the cholinergic system, which plays central roles in memory encoding, attention, and cognitive flexibility. Alzheimer's disease is characterized by progressive degeneration of cholinergic neurons, the neurons that produce and respond to acetylcholine. The most widely used pharmacological treatments for Alzheimer's disease, the acetylcholinesterase inhibitors, work by blocking the enzyme that breaks down acetylcholine, effectively compensating for the reduced production by damaged cholinergic neurons. Dietary choline does not reverse Alzheimer's disease, but research on choline intake and cognitive aging suggests that adequate choline throughout adult life supports the long-term integrity of the cholinergic system. A study published in the American Journal of Clinical Nutrition found that higher dietary choline intake was associated with better cognitive performance, specifically better verbal and visual memory, in a large population of adults. Research from the University of Eastern Finland, published in the American Journal of Clinical Nutrition, found that men with higher dietary choline intake had a 32% lower risk of dementia over a 22-year follow-up period. The association held after adjusting for other dietary factors, lifestyle variables, and known cardiovascular risk factors. 2 eggs contain approximately 250 mg of choline, more than any other commonly consumed food. The concentration of choline in eggs is exceptional. The next best dietary sources, liver, fish, and certain legumes, are consumed far less frequently in most diets. For the majority of people who are not eating organ meats regularly, eggs are the most practical dietary route to meaningful choline intake. The choline is in the yolk. Egg white omelets, the response to decades of dietary cholesterol warnings, eliminate the one component of the egg that provides the most nutritionally distinctive and irreplaceable benefit. The fat in egg yolks deserves specific attention because it is the component most implicated in the original dietary fat hypothesis and the component whose actual effects are most misunderstood. Egg yolks contain approximately 5 grams of fat per large egg, distributed across saturated fat, monounsaturated fat, and polyunsaturated fat. The monounsaturated fat is primarily oleic acid, the same fat that predominates in olive oil and that is consistently associated with cardiovascular benefit rather than harm in the research literature. The saturated fat in eggs, primarily palmitic and stearic acid, has different metabolic effects. Stearic acid, which makes up a significant portion of the saturated fat in eggs, is converted in the body to oleic acid rather than raising LDL cholesterol in the way that shorter-chain saturated fats do. The saturated fat profile of eggs is categorically different from the saturated fat in, for example, partially hydrogenated vegetable oils, and treating them as equivalent was a significant error in the original dietary fat hypothesis. Egg yolks also contain lutein and zeaxanthin, carotenoids that accumulate in the macula of the retina and protect against age-related macular degeneration, the leading cause of vision loss in adults over 60. These carotenoids are fat-soluble, and their absorption from eggs is significantly higher than their absorption from plant sources because the fat in the egg yolk facilitates their transport across the intestinal membrane. Vitamin D, present in egg yolks in meaningful amounts, is another fat-soluble nutrient whose absorption from eggs is enhanced by the egg's own fat content. Most foods contain little or no vitamin D. Eggs are one of the few non-fortified food sources that provide meaningful amounts. The practical question of how many eggs constitutes a beneficial daily intake has a clearer answer from the research than most people expect. The studies that showed no association between egg consumption and cardiovascular risk typically examined intakes of 1 to 3 eggs per day. Some research suggests that going beyond 3 eggs per day may shift the risk profile in certain populations, but the evidence at this level of consumption is less consistent. The cognitive benefits associated with choline, the most documented nutritional benefit of eggs, are observed at intakes that 2 eggs per day reliably provide. 2 eggs deliver approximately 250 milligrams of choline, a significant contribution toward the recommended intake that is difficult to match through any other commonly eaten food. The practical recommendation that emerges from the research is that 1 to 2 eggs per day as part of a varied diet does not increase cardiovascular risk in healthy adults without diabetes and provides meaningful amounts of choline, vitamin D, lutein, zeaxanthin, and complete protein that are difficult to obtain from other sources in comparable concentrations. The preparation method matters for some of these nutrients. Cooking does not significantly affect choline content. Choline is heat stable. Vitamin D is also relatively stable in cooking. Lutein and zeaxanthin absorption may be slightly higher from cooked eggs than from raw eggs because cooking disrupts the protein matrix that encapsulates them, making them more accessible during digestion. The optimal preparation from a nutrient preservation standpoint is gentle cooking at moderate temperatures, poached, soft-boiled, or lightly scrambled, which preserves the integrity of the fat-soluble nutrients without the oxidation that high-heat cooking can produce in the polyunsaturated fat components. Here is the synthesis. For 40 years, public health guidance told people to limit eggs based on the dietary cholesterol hypothesis. That hypothesis was wrong for most people. Dietary cholesterol does not meaningfully raise serum cholesterol in approximately 70 to 75% of people. In hyperresponders, it raises both LDL and HDL without unfavorably shifting the ratio, and the LDL particles that increase tend to be the large, less atherogenic subtype. Large prospective studies following hundreds of thousands of adults for decades do not find associations between moderate egg consumption and increased cardiovascular disease in healthy populations. The 40-year warning was not supported by the evidence that accumulated after it was issued. Eggs are the most concentrated common dietary source of choline, a nutrient that most people consume at less than 60% of the recommended intake, and that is essential for acetylcholine synthesis, memory encoding, and the long-term integrity of the cholinergic system associated with Alzheimer's disease risk. The fat in eggs is primarily oleic acid and stearic acid, fats with neutral to beneficial effects on cardiovascular risk markers, along with fat-soluble carotenoids and vitamin D, whose absorption is enhanced by the egg's own fat content. Everything else. 1 to 2 eggs per day, whole, gently cooked, is supported by the current evidence as a health-positive dietary practice for most adults. If this changed how you think about the eggs you have been limiting for years, hit the like button. Share it with someone who switched to egg whites because their doctor told them to limit cholesterol.